next speaker is Dian Lyu from Stanford University, and the talk will be about intracranial electrical stimulation. Let's welcome the speaker.
Hi, everyone. Thanks for the organizers for inviting me to give the talk, and thank you for the audience for staying so late for my talk. So today I'm going to share some of the recent findings we have about thalamo-centric causal connectivity mapping in the human brain with intracranial electrical stimulation. So we know that the thalamus is very important in shaping the global functional architecture of the brain. But most of our knowledge about the thalamus comes from animal studies, because in human brain, we lack the tools with enough spatial temporal resolution to have access to subcortical areas. But now the opportunities are changing, because since 2018, the deep brain stimulation of the anterior thalamic nucleus has been approved for treating epilepsy. And many of the clinical teams in the world, including our team, begins to do a multi-site thalamic recording from a single patient. Because with intracranial EEG, we are dealing with sparse recording. In order to analyze how different nuclei in the thalamus are differentially connected with the rest of the brain, it's very important to do it with the same brain. Even though in the previous literature, it has always been considered as a gold standard to use brain stimulation to map human brain function. But as a matter of fact, the direct stimulation effect is really complex and understudied. So in the presentation I'm going to give today, first I'm going to present the method we are going to tackle with the problem, how to quantify and characterize the complex profiles of the evoked potential. Once we quantified those features that define the evoked potential, we are going to use those as a template and map out in individual cases of stimulation and recording sites, when and where do those features occur given the stimulation. So. This is our electrode localization. So at the left, you see the electrodes at the group level span across the whole cortex. And in this grouped situation, we have 27 participants with about 5,000 electrode contacts inserted across the brain area. And at the right, we have thalamic recording and stimulation sites. And we roughly differentiated the thalamus into 3 different nuclei, ant, mid, and posterior. So ant roughly corresponding to ant, but it also has some of the ventral anterior nuclei. And posterior nuclei, we know pretty sure, because it's a large structure, is a pulvinar. And in our method, our research team has been systematically performing the procedure of repeated single electrical pulse stimulation on every recruited patient. So in this procedure, we stimulate every available contact while recording from all other sites. In this way, we can map out the pairwise connectivity based on what contact we have. So in the first part of the study, we need to define what activation means. So roughly use the word activation basically means significant evoked potential based on a stimulation effect. And because it's a very complicated situation where you have different profiles, but some of those, by eyeballing them, we can list some of the criteria that are physiologically plausible that to be a evoked effect transmitted by the axons rather than the volume conduction. So we use those human knowledge to define the criteria, and use the semi-supervised learning to extract those features of the evoked spectrograms. By the way, so the data I'm training here is power and intertrial phase coherence at spectral the spectral information instead of the time information. And then you can use the semi-supervised learning based on human knowledge to characterize all of the data into two clusters. One is the activated cluster, and another one is non-activated cluster. And then you do a subtraction to get a clean cluster of the activation defining features, and then those data sets will enter into group level analysis. And then we use some other nonlinear data dimensionality reduction methods to further cluster those activated pairs into 4 different spectrograms. And it turns out the 4 different spectrograms are driven by 3 features. And these 3 features, spectral features, are as follows. The F1 feature is characterized by a peak power at the gamma band, high frequency band, in a very early onset time. So it's upon stimulation within 60 milliseconds, there is a high peak. In time domain, as a time domain, it corresponds to the first peak, like high peak, and one component in the time domain. And the second feature corresponding to a lower frequency peak at the power band, and it doesn't have a phase locking feature, which is very different than the third feature we are discovering here, which also has a low frequency peak at the power spectrogram, but is more delayed than F2, and it has a very strong narrow band phase locking feature. At the time domain, it shows something resembles a theta oscillation. And most importantly, this F3 feature is uniquely associated with thalamic evoked responses, which is unseen from all other cortical evoked responses. So using those group level results, so those group averaged results, we can see it as a template of a feature. And we use that as our reference and do a cross-correlation in each individual spectrograms of specific size of stimulation and recording area, and by which we construct this causal connectivity matrices using the feature representation. And as we can see, the F1 matrices show a very striking modularity, by which that we see regions in the same hemisphere and in the same lobe are more closely connected than the rest. But thalamus is very interesting here because it receives bilateral widespread sources of cortical influences. F1 feature, however, is more spread out than F1. F2 feature is like expanded by F1, but remain this modularity. It seems to say F2 connectivity is indirect connectivity built on top of the faster feed-forward F1 connectivity. And what is very interesting is the F3 connectivity is uniquely associated with thalamus stimulation. So from the row, you see the stimulation sites. From the column, you see all the recording sites. By stimulating thalamus, you are going to record the widespread theta oscillation in bilateral cortical areas. And because there is a very nice symmetry of the F1 inflow pathway of the thalamus and F3 outflow pathway of the thalamus, we naturally ask whether they are recurrent connectivity, they mirror each other. So we did a comparison of the thalamocortical F3 versus cortical thalamic F1 pathway. It turns out that it depends on which region you are looking at. For high-ordered cortices like frontal parietal control network and default mode network, those regions have much more expanded F3 representation than the inflow connectivity, which probably means that for those high-level cortices, the influence of the thalamus expands away from what it receives. So it may involve a lot of cortical loops in those areas. So next, so there is some misrepresentation of the slides, but it's okay. We are going to focus mainly on the first panel. So here we can look at the time information about when those features occur in different sites. And we could see, based on the histogram of each category of the time information when the feature occurs, that the earliest influences you can receive upon a cortical stimulation is thalamus. So thalamus not only receives widespread cortical connectivity, but it also receives the earliest. And it is similar to the other direction. When you stimulate thalamus, and the F3 oscillation happens the earliest in the cortex regions. So we also saw from the previous matrices that the cortical regions can have some theta oscillation, but those are later than thalamo-cortical oscillation, which probably means those oscillations are derived from a thalamo-cortical. So this is the end of my talk, and do I have my...
Okay, so we got one question in the app. The question is, in F1, is there a possibility that there is artifacts or crosstalk from the stimulation?
Artifact is what kind of artifact? So there is electrical artifact, and in the method section, we actually dealt with by subtracting. So based on the semi-supervised learning, we also label what is absolutely not an activation, but an artifact. So in the semi-supervised learning, you also need to learn to dissociate those cases. And those cases will be grouped into the non-activated cases. And before the data enters into group level analysis, the artifact will be subtracted from the activated spectrograms. And the other thing to avoid the artifact is that we know if the strong evoked potential, or the strong peak in the time domain, if it happens very early, like within the 10 milliseconds, it must be not must, but it most likely is an artifact. And all of the analysis is done after 10 milliseconds. So that's another way we avoid that our result to be an artifact. And there are some other quality control things that I don't have time to expand, but it is indeed a very important point to make that, especially the F1 feature, the early feature, is not an artifact. If that's an artifact, what he meant. But if there's some other artifact, I'm not sure. So. And the other possibility.
We have a question here at the front.
Yeah,
for
sorry.
you.
Hi, this is fabulous work. So you're showing that there's two different ways that you can look at the evoked potential with power and with ITPC, and I was just wondering, as you move forward, did you find that the most significant results were using more of the power or the phase-based analysis?
So the feature here are a combo of two.
Combo, okay,
yeah.
Interesting.
Yeah.
And I can see now that, since I'm getting to look at it a second time, that you had specifically mentioned theta as the frequency band that was most salient. Was that kind of like a data-driven process, or did you kind of look at, was it just like the peak in the power spectral density, or did you choose canonical frequency bands and go from there?
So yeah, it's a data-driven thing. So I didn't specify what canonical frequency here. I used the whole spectrogram, and it turns out to be most highlighted in the theta band, and that band seems also to differ in different subjects, but at the group average, it's about 5.
Awesome. And sorry, last question. Did you ever find like suppression of activity to be localizing as well, or like interesting?
So in our method, we are unable to differentiate suppression or excitation.
You can go both ways? Is that what you mean?
So we are unable to differentiate excitatory or inhibitory effect.
Ah, okay. Yeah, you're just looking for the
so variance.
Either way.
The variance comparing to the baseline.
Got it. Thank you.
I think it's really fantastic work. One of the things that I was thinking, I mean, the matrices that you showed there, the F1 to F3, they have really a clear structure in them. Have you given thought to perhaps looking at whether these can be predicted or associate with, for example, structural connectivity that we would infer from tractography? I mean, one possibility, an exciting possibility, would be that you would use your F1 to F3 to actually infer the directionality of a structural connection if you're to
Exactly.
combine
So,
them.
yeah.
So I wonder if you could speak a little bit about future work that you can, where you can take this, because this seems to be a really exciting
Yes,
approach.
I agree. This is exactly what I was thinking of, one direction it could go into. So it can have a lot of influence in both the animal work to be more molecular, like mechanistic, why the oscillation happens, what things is going on in the local circuit level that make it happening. Because it's single pulse. What we are stimulating is single pulse. It's just one single pulse, and then it causes this oscillation. It must be some loop going inside. But on the other hand, we can look at the global structure, it's what you said, to take into account the structural connectivity based on DTI and do some simulation work to see if we can recapitulate the causal connectivity pattern that we see here. So this, yeah, it's very exciting.
That would be really cool.
Yes,
I'd love to collaborate with you on that if you...
Thank you.
Yeah.
Thank you.
Are there any other final questions for our speaker? Please join me in thanking the speaker.
Thank you.